I'm Ben Raphael. It's a pleasure to be back at CGSI for the nth time. I don't know if I've lost count, but this will be something a bit different, something new for us, tracking development over space and time. So I think one of the biggest scientific questions, probably of all time, is how does a single cell become a complex multicellular organism? More specifically, what are the molecular changes that occur as the single cell zygote divides and differentiates into the trillions of cells that make up, say, a human or other mammal? Now, what I'm going to do is tell you about some of our recent work on this very large question, but I should really start with two caveats. The first is that I'm not a developmental biologist, so you should double check anything I say by asking ChatGPT. And second, this is my first time presenting this material. This is new stuff for us, so maybe a little bit rough, but we'll see how it goes. So, development, I think, to sort of a high level, can be described as a superposition of two processes. Cells dividing can represent cells dividing via a tree, a binary tree. One cell divides into two. And cells differentiating. And in developmental biology, they often describe this differentiation process through this classic Waddington landscape, where you can think of the multipotent cell, the cell that hasn't yet differentiated, is represented via this marble, which rolls down this epigenetic landscape. And this epigenetic landscape has these valleys that represent favorable epigenetic states. And it reaches sort of the end here. There's some terminal cell state, which is sort of a stable cell state. So this is the original image from Waddington's classic 1957 paper, but you'll see variations of this all throughout the developmental literature. I mean, here's another one where they're just sort of illustrating different neuronal cell types that are differentiating as the cell sort of moves down this epigenetic landscape. So we could think of three key questions in terms of this simple view of development. First, what is the history of cell divisions over time? Computationally, we could just say that's the problem of reconstructing the cell lineage tree. What are the cell types or cell states at each developmental time point? And so we could think of that computationally as grouping cells according to an epigenetic state. And then what's the history of cell differentiations over time? And we can also describe this as a structure inspired here by the Waddington landscape that I'll call the cell differentiation map, and I'll talk about this a little bit later. And so these three key questions have been answered mostly for really only one higher order organism, and that's C. elegans. And through a set of really classic and laborious experiments, Salston and Horvitz and Sidney Brenner won the Nobel Prize for deriving the complete lineage tree of C. elegans, all 959 cells. Here's the whole tree, sort of zooming in on one part. And they sort of determined the developmental fate of every cell and identified key genes that control important processes like programmed cell death and organ development. So we'd like to do this for other organisms, but this sort of manual, laborious process won't scale, certainly won't scale to trillions of cells. We need high throughput technologies. And fortunately, as you know, we have such technologies. There have been tremendous advances in single cell and spatial RNA sequencing. So this gives us a means to measure something about cell state in high throughput. And there have been other technologies, genome editing technologies, that allow biologists to induce mutations in genomes, thereby giving a marker of cell lineage, of cell division. And so what I want to talk to you about today are two projects that we've worked on to address parts of these key questions. The first part using RNA sequencing, and the computational framework is optimal transport. And then in the second part, using technology of dynamic lineage tracing, and the computational methodology there will be classic phylogeny. So beginning with the first part, thinking about RNA sequencing, I'm sure many of you are familiar with single-cell RNA sequencing, technologies that isolate single cells, tag them, barcode them in a high-throughput way, and measure gene expression for all or most genes in thousands, tens of thousands, in some cases millions of cells now. And so there's a whole series of data processing that happens, and I'm not going to talk about any of that. And we could just think in simplicity, after all of this careful data processing, that what we can obtain from the single-cell RNA sequencing is a measure of cell type or cell state, represented here by clusters in this two-dimensional visualization. And then the other sort of way of analyzing single-cell RNA sequencing is not to identify discrete cell types or cell states, but to do what's called trajectory inference and look at sort of the continuum of changes in state between cell types or between intermediate cell states as a system goes through a developmental process. So this is, again, a really deep field with lots of methods, but what we can think about doing is inferring some sort of branching structure describing the differentiation of cells into different cell states. So this is this idea of this differentiation map. So there's many techniques for trajectory inference. One of my favorite, and the one I'll sort of describe and build off of in a way, is one called Waddington-OT, which it attempts to actually really model this idea of this Waddington landscape by describing it as a map between probability distributions. And so this is a figure from the Waddington-OT paper from 2019. And what they sort of show here at sort of the initial time t1, they measure cell states, and it's this unimodal distribution. So maybe there's sort of a single cell type and some variation around that. And then over time, at a later time, what you observe is the distribution of cell states sort of bifurcating. So maybe at this later time t2, we see this bimodal distribution of two cell states. Okay, so this is sort of a mathematical description of the Waddington landscape, although I should note here that the figure, just so you don't get confused, time is going upward in this figure, and it's going sort of downward in the Waddington landscape. So just to show you a bit about the mathematics, because we're going to build on this idea of finding this map between probability distributions is something that's been well studied in mathematics for centuries, actually, going back to the 18th century, and has actually recently become popular in machine learning and also become popular in single-cell analysis. And it's a framework called optimal transport. And so the idea is to find optimal maps between probability distributions. So if we think about a probability distribution as, say, a pile of sand, if we're down at the beach, we want to sort of transform this pile of sand into this shaped pile of sand. This is our unimodal distribution and our bimodal distribution. And there could be many ways we could sort of move sand around, but we want to do this in a way that minimizes the amount of sand we move. Right? We want to minimize work. And so what that means mathematically is we write down a cost function that tells us the cost of moving sand from location I to j. So we have some cost function. This could just be distance between points I and j. And then we try to minimize the total cost of moving sand according to our map. How much sand do we move from I to j? Okay? And we just sum over all such pairs, and we try to find the optimum such map. Okay? It's a very sort of straightforward idea. But there's also these constraints. And what these constraints tell us is that we want to sum over all the map. We should get all the sand in px should be moved to py, and all the sand from py should have come from px, right? So we're sort of moving everything from one to the other. So this is a really beautiful framework, and it's been extended in various ways. But there's one key thing missing about development in this framework. And the key thing missing is that development doesn't just happen in this abstract cell state space. It happens in actual physical space, right? Cells divide, they occupy space, and although, in the first few stages of an embryo, they might be sort of, amorphous, there are dramatic changes that happen in creating the structure of the organism, right? And so, there's an early stage in development called gastrulation, where the body plan is sort of laid down. Okay, so this Waddington landscape, has no notion of physical space, right? The space here is this abstract epigenetic space. So let's sort of do a thought experiment here. We think about this map that's being described. Let's imagine a one-dimensional organism. Maybe you've read the classic book Flatland, where they have these one-dimensional organisms. And so I could take this notion of cell state, and now let's put for each cell a location. So it's just a location in this one-dimensional coordinate. And so here I've done that. You can see I put all the, the cells at that first cell state in the center, and the other ones are here. So there we are. I've just added each cell the location. And now what happens to this optimal map when we look at it now in abstract cell state space and in location space? And the map looks like this. So cells have moved from one time point to the next, and these lines are indicating how they moved in this space. Now, this is the optimal map in terms of minimum amount of movement in expression space. But now if we look at these lines and we think about physical space, you can see that there are cells moving from the bottom of the organism all the way to the top, and vice versa. So there's all this sort of movement of cells that have happened, which doesn't seem realistic at all. So instead, we ask for a map that views both expression space and physical location. We can derive a map like this. Okay? And so this map, you can see actually no cell moves in its y-coordinate. So there's no movement in physical space, and there's movement only in expression space. Now, it turns out that the total sum of the movement in expression space is slightly more than it is here. Right? So there's a little bit of a trade-off. But I would argue that this seems like a much more physically plausible differentiation process than the one without considering space. Okay, so that's pretty abstract, right? It's like a nice sort of thought experiment. But how can we actually make this real? Can we actually do this? So fortunately, single-cell sequencing technologies are being rapidly replaced by spatial sequencing technologies. And in particular, single-cell RNA sequencing is being sort of supplanted by technologies called spatial transcriptomics. So the idea here is that you can take a 2D tissue slice, and you can measure simultaneously from thousands of locations in that slice a high-dimensional gene expression vector. Okay, so the data you get is all genes by thousands of spatial locations, and you've recorded where in 2D space you've made that gene expression measurement. Right? So this is this extra piece of information, this space, that we didn't have before when we were viewing single-cell RNA sequencing technology. Well, so there's many technologies, and I guess I'm giving a tutorial, so I might talk a little bit about some of these technologies on Thursday. And they have different resolutions and different number of cells, et cetera, and let's not worry about that for the purposes of this talk. But the interesting thing in thinking about development is we could apply these technologies, and now we can apply them not at one time point, but we could apply them at multiple time points during development. So we'll call this spatiotemporal transcriptomics data. Now, I should say it's a destructive technology, so we have to have different animals when we apply the technology. But if we assume the developmental process is mostly conserved, we can start using these time points and sort of looking at how cells differentiate across this gene expression and spatial axes. So key questions: can we identify the ancestor-descendant relationships between adjacent time points? Which cell was the ancestor of which cell at a previous time point? Which cells or cell types are growing? Which are not growing? Which are in fact dying? So programmed cell death is an important part of development. What are the rates of these things? And ultimately, what are the gene programs that govern proliferation, cell growth, and apoptosis, cell death? So to answer some of these questions, we developed an algorithm that we call DeST-OT, or I guess it's supposed to be destiny, kind of. And this is joint work with Peter Halmos and Julian Gould, who are here and can answer more questions about it, and Jinhao Liu, who is not here, unfortunately. And so what does DeST-OT do? So given the spatial temporal transcriptomics data, it computes an alignment between pairs of spatial transcriptomics data, pairs of time points. Maybe I'm using my hands too much or something. It's going to allow us to estimate growth rates in an unsupervised way, which I'll show you. And it's going to provide some insights into the gene expression programs of growth and differentiation. So to dig into this a little more, to describe some of the techniques behind this, it's built on an earlier work from my group called PASTE, which I described at CGSI actually a couple of years ago. PASTE was designed to align spatial transcriptomics data sets from multiple adjacent spatial slices. So the idea being that tissues are 3D. Spatial transcriptomics gives you a 2D slice of a 3D tissue. So if you had taken multiple slices from this tissue, you'd want to align them back together and align them such that you could build back in 3D the tissue, or you could, in fact, integrate the slices together. Thinking just about this sort of pairwise alignment, here's sort of one example of two adjacent slices from the human prefrontal cortex. You can see this sort of layered structure. These colors indicate different layers of the cortex, and the alignment here is extremely good because these are two adjacent slices in space. And so how does it work? So it uses this optimal transport technique that I talked about, moving these sand piles. But now it needs to move these sand piles, thinking not just about similarity in gene expression space, but similarity in physical space. And the way we do that is we just add a second term to this objective function, to the thing that the optimal transport is trying to minimize. So this first term is the one you saw. This is what's the cost of moving sand from one place to another in gene expression space. And then in physical space, you actually think about what's the distance between pairs of aligned points. Okay, so you actually sort of do this. If you're going to align this pair of points to this pair of points, then you look at they should have similar distances to each other. And this is a framework called fused Gromov-Wasserstein optimal transport. So it's sort of a mouthful, but it's really just a convex combination of these two terms. And you have, again, these balanced terms, which tells you that all the spots should be mapped. And there's some nice features of this that I won't sort of go into now. What I wanted to talk about is actually this sort of constraint right here about balance. Because since we introduced PASTE, a number of groups have extended this in various ways. We ourselves extended this using what's called partial optimal transport. So this was PASTE-2. And these constraints about all the sand has to be moved, we relaxed. So this was to allow us to allow only align a fraction of the slices. And this was important because, especially in 3D, when you sort of cut an ellipsoid, for example, one slice might be smaller than the other, so you don't want to align all the spots. And then recently, while we were working on DeST-OT, another group sort of came up with a method that actually used what's called unbalanced optimal transport. So they get rid of these balance constraints and replace them with these soft constraints, where they allow imbalance in the alignment. So here I'm showing these histograms. The balance is that the two things are the same. Here there's this imbalance, and they add these KL divergence terms to give you these soft constraints. So what can happen? So when you apply these things to development, and our original idea was just to use PASTE or PASTE-2, what we quickly learned is that development is neither balanced nor is it unbalanced. Okay, so balanced would mean that there's no cell death. You have to map every cell from one time point to the next time point. Unbalanced, it turns out, is just completely unconstrained in terms of growth and death of cells. And it doesn't really respect anything about kind of the arrow of time that happens in development. So neither balanced or unbalanced we found to be very good. And instead what we used was an intermediate called semi-relaxed optimal transport. And this actually is a framework that existed. It's not very widely used, but it's, in fact, the very natural framework to use for development because it models a natural asymmetry in the data, which is every cell at the later time point in development had an ancestor. Okay? So if you think about these histograms, every cell from the later time point came from somewhere, right? But the cells at earlier time points may have multiple descendants, they may grow, or they may have no descendant. Okay? So you only want the sort of balance in one direction. And more interestingly, the difference between these two marginals gives you the growth rate. The growth, or if it's positive, or death if it's negative. Okay, so this is a sort of growth vector. Now, I know this has been a pretty abstract talk to this point, so let me try to give you an analogy for these three different optimal transports. So I'll give you maybe the analogy for the Los Angeles audience. Okay, so balanced optimal transport are like these East Coast bankers, not very relaxed, really constrained. Unbalanced is like maybe this guy you'd see on Venice Beach A little too relaxed. Maybe should occasionally work a little bit. And then semi-relaxed is like this guy you might see in Santa Monica when we're there, or Malibu, or Hollywood, right? He works, but, he looks pretty relaxed. So semi-relaxed is really the way to do it, okay? Now, there's something else about DeST-OT with the objective function. We actually, to allow growth or death, we actually have these triplet terms in the objective function. We allow pairs of points to map to singletons, or singletons to grow into pairs. I won't really describe that. It's in the preprint in the paper, and you can go sort of read that. So putting it kind of all together, the important things are, there is this objective where we allow these extra terms, but this semi-relaxed, this asymmetry in development is really important, and we get these growth rates. And then we can just sort of solve this using a variant of the classic Sinkhorn algorithm. Now, CGSI is not really, kind of a sort of a methods and not results, but I'll just show you a few results just so you sort of see that there's something going on here and it's not all just a bunch of equations. So here's one data set where, from our collaborator leading at WashU, this was kidneys from mice for multiple weeks of life here. You can see that the kidney grows. Actually, it goes off the slide, grows bigger than the slide. And the colors here are their cell type annotations. And so we can analyze, we could compare different methods, and we can see how much do these methods preserve gene expression, how much do cells move between adjacent time points. I'm not going to go into all the details. I'll just tell you we do the best. Read the paper, okay? But what we also do really well is we get these growth rates, right? This semi-relaxed gives us this difference, so we get this growth rates. And if you look carefully at this, you'll sort of see from one time to the next, this pink, for example, you can kind of see there's more pink cells in the week 12 than there were in week 3, so there should have been growth in pink. And this heat map is actually our inferred growth rate, so you can kind of see this bright green is the highest growth rate. Later, here's this like lavender. You can see kind of the lavender increased from 12 to 52. And we can see these high growth rates. And then this unbalanced mascot, it just has death everywhere, and it's like the whole kidney is regrowing each week from a small number of cells. It's really bizarre. So this looks much more plausible, we would argue, than this. All right, one other result. Here's a different spatial technology called StereoSeq. It's a subcellular resolution, lots of cells. These are aligned. These are brains from different weeks of development. Axolotl. I think actually we heard about it in Ryan's talk. So it's this, it's a really cute species of salamander. It's so cute that they make oversized plush toys for kids, like dramatically oversized. But it has actually really interesting biological properties like regeneration and stuff, which is why they were sort of analyzing it. Because this technology is such high resolution and has many spots, DeST-OT will run on this scale, but it sort of motivated us to think about scalability. And I should say what I'm about to show you on the next slide, there was one reviewer at the International Conference of Machine Learning that did not want you to see this next slide. Okay? So the submission was rejected. However, 4 of the 5 did recommend it, and this was our way of finding a low-rank approximation of this map. So it's a way of scaling by finding a low-rank approximation. And so it's actually quite amazing, and Peter and Julian can tell you more about this. So this full-rank map, here it's P instead of pi, sorry about that, you decompose into these lower dimensional optimal transports with this latent coupling. And this latent coupling gives you kind of this low-rank approximation of the full coupling. So from these alignments, this is just another view of what we can do. These are deriving, using their cell type annotations, the descendants of each of the cell types from the previous time. So here's the earlier stages, the colors here are the previous times. Maybe you can't see it, maybe you can. Oh, the monitor's pretty big. So each of these arrows describes an ancestor-descendant relationship between different cell types at the different stages. So this is this idea of deriving the cell differentiation map. And we can do this directly from the output of the DeST-OT, directly from this map we get. And it recapitulates many of the known cell differentiation. It has some putative new stuff. Some of the ones are things that were in the literature that people postulated, and we find them there. And now, Peter, who's there, really wanted this slide to be shown. So I'm going to show you sort of this next slide, which is that we can also do interpolation between the time points. So this is actually a much larger data set, this stereo-seq technology of mouse embryo. And what's being shown here is the alignment from these two developmental time points, but then interpolated in the intervening time. And these are just sort of two different views of the gene expression. So it's a pretty picture for sure. And we're looking at what we can kind of do with this. All right, so that's DeST-OT. That's the first part. Sort of align the spatial temporal transcriptomics data. Semi-relaxed OT. Semi-relaxed is the way to go because you get these growth rates, you get these cell type transitions. And I didn't really tell you about these sort of metrics, but this idea of looking at how much do cells move in both gene expression space and physical space. And it's important to look at both when you're doing spatial temporal alignment. And we can do some benchmarking, and it does pretty well. All right, so in the last part, I think I still have a little bit of time. I'm going to switch technologies. I'm going to switch everything. Right? The only common theme here is going to be development. So what about reconstructing the cell division history and this differentiation map? So this is going to use a completely different technology. This is a technology that induces mutations into a developing organism. Okay, so what we want to do here is we want to figure out what's the history of cell divisions during the organism's growth. So that we need a marker then of cell division. So mutations actually naturally occur in the genome. These are somatic mutations, but they typically occur at pretty low rates. And you don't know where they're occurring, so you'd have to sequence the whole genome to find them. Right? So in order to get around these issues, you can now introduce mutations in the genome at prescribed locations through CRISPR and other such techniques. And then you can read those out with single-cell RNA sequencing. Okay? So that gives you both a measure of the mutations, you sequence the mutations, and you sequence cell state, because it's a full gene expression measurement. So taking this data, there are sort of two fundamental problems. The first one is just to get back the cell lineage tree. So infer the cell lineage tree from this mutation data. This is just a classic phylogenetic tree inference problem. However, because there's there's particular error rates here, and there's actually sort of particular evolutionary constraints that you can use because of this experimental system, you can develop specialized techniques for this. Matt Jones, who's sitting right here, actually was one of the pioneers in this, in developing Cassiopeia when he was in Nir Yosef's group. We've worked on this a bit. And we have two tools. One, Startle, it's developed by Henry Schmidt, who's here somewhere at CGSI. Another one, Lamo, with Ewan Ma, who's here as well. And they can tell you about sort of what they've done there. But the next problem is once you have cell division history, what you'd really like to know also is this differentiation map, which I've kind of alluded to so far throughout the talk, but now I'm going to define very precisely. So the cell differentiation map is a directed graph where the nodes are cell types and directed edges indicate differentiation events. When does one cell type become differentiated into another? Okay, so that's what you'd like to know. That's a really important part of developmental biology. What are the progenitors, the ancestral cells in differentiated space of the terminal cell types? So how do we get that? Well, if we knew the cell types of all the ancestors in this tree, then we could just travel down this tree and see every time a color changes. So I've used colors here to do cell states. Every time a color changes, there was a differentiation. Okay, so I could get this differentiation map directly. Of course, the challenge here is that when we do this lineage tracing, we have done a single cell readout at the end of the process. So we only know the cell states of the leaves of this tree. Now, if you've taken a reasonable bioinformatics course, like maybe one that used Pavel's book, you know how to solve this problem. If you have measurements on the leaves of the tree, you want to infer the states on the ancestors, and you want to do this in some optimal way. So you could certainly try to find the most parsimonious labeling of these ancestors. By that, I mean the fewest color changes, the fewest differentiations. Or you could use a probabilistic model and find the most likely ones, right? There's Fitch's algorithm, Sankoff algorithm, classic algorithms for do this. Okay, so we might do that here, and we would obtain this differentiation map. But actually, that wasn't the one I showed you a couple slides ago. And what happened? I'll show you it again on the next slide. But here, our differentiation map only includes the states at the present, because that's all we know. So we get these differentiations that actually don't make really any sense. Okay, so what we wanted to get was this. But that's sort of bizarre because we don't have the purple color cell and the blue cell in our data. They were unmeasured. So now we have to think about sort of different problem. We observe these cell types at the present time. Some of them were progenitors back, further back in developmental time. Some are terminal cell types. There's sort of things here. Some of the progenitors we measured at the present time, some we didn't, and we don't know. So what can we do here? This is actually a kind of fun, interesting problem. And the state of the art is that if you observe all the progenitors at the present time, then you can do Sankoff the Fish algorithm, the one I described. This is also implicitly what people are doing in trajectory inference from single-cell RNA data, because they're assuming they measured all the progenitors and they're connecting them in between. If you don't observe any progenitors, if you assume that none of the progenitor cell types are observed, then there has been some work in lineage tracing that just uses distance-based phylogeny methods, and they build these binary trees, and it's sort of not quite the cell differentiation map, but it's something. And then there's this big empty space in between. Okay, and so this big empty space is what we have filled in a bit with our method called CARTA. So CARTA is not an acronym for anything, so I don't know why it's capitalized here, but it infers cell differentiation maps from lineage trees. Okay, so the idea here is that you input a lineage tree. Again, you're only going to measure these cell states at the at the leaves, and then somehow you infer the differentiation map, which you output. Okay, and what CARTA does is it explores, and if there's time, I'll describe the sort of trade-off between how many unobserved progenitors do you allow and how closely do you want to match the observed trees. And it turns out there's this really interesting trade-off between these two objectives. And now, some of you, I know there are a few aficionados in the audience, and the aficionados might look at this and remember another paper. One person in this room in particular might remember one of the papers from his postdoc when there was a kind of similar trade-off when we were looking at metastatic cell migrations and metastases. And there was this idea of this trade-off between how complicated the migration map was and all these sort of things. So maybe some of you know this paper or know this sort of idea. And it seems like vaguely similar, but it's actually not quite the same trade-off. Right? So the missing thing here, the thing that actually is going to allow us to get around this sort of issue of the unobserved progenitors, is that this is development we're dealing with. And in development, progenitor cells differentiate into terminal cell types, or they differentiate into other progenitors. So there's a sort of hierarchy from the sort of totipotent progenitor, maybe the zygote that can make any cell type, into increasing series of restricted progenitors. And we could describe any progenitor by a set of cell types that its descendants can attain. So progenitor is just a set of cell types, what we call the potency set. Okay? So that's actually sort of the key thing. And so then what we really want to do is, if I gave you this map in terms of now these sets, all I've done is I've changed that color. That purple color that I didn't know is now just the potency set, all the cell types that I can differentiate into, and I can label the cell lineage tree this way. But what we want to do is with the reversed, we want to find this cell differentiation map that best fits the data. Do I have time? Should I describe this? I feel like at least by starting clock, I have time, but I know people want to get to the beach. All right. So here's a trivial solution. Anyone have like a completely trivial solution to this problem? Here's the most trivial differentiation map around. You just create what's called the totipotent progenitor, meaning it produce any cell type. Okay, so I look at all my observed colors here, my cell types, and this is the progenitor can produce anything. And that's a cell differentiation map. And now I can then take this map and I can go back and label the ancestral nodes using this map, which means I get to choose from this set that can produce anything, or I can produce any of these colors, and here's sort of the minimal number of changes I'd have to make for this map. Okay, and now what's kind of weird about this labeling? Okay, what's strange about this labeling is that if you look along the tree, you might see like right here, this part I'm circling, we had this totipotent progenitor that could have produced any of 4 cell types, but it only actually produced these 2 cell types. So it didn't produce the others for some reason on this tree, right? So that seems kind of weird, right? Why only did it 2-2? So what we want to kind of quantify is these ideas of the discrepancy, okay? And so here, if I were to sum up how many unobserved cell types that the progenitor could have produced but didn't, the total discrepancy here is 7, okay? So then there's another quantity, discrepancy 7. There's only one progenitor, right? The totipotent progenitor. Right? Now on the other extreme, what could I do in the other extreme? I could just introduce as many progenitors as I want. Okay? If I produce as many progenitors as I want, what that corresponds to is just take the union of these sets as I just go up the tree. Right? So here it's 2 red, so this should be red. Green here is a, red and green, so this should be red and green. Sorry for colorblind, this is red and green. There it is. This is like what actually has happened in Fitch's algorithm, right? You put these sets together. So this is now a differentiation map. It's got discrepancy 0 by the way we constructed it, but it's got a lot of progenitors, 5. And so now we have this trade-off. And so now we can think about how many progenitors do we want, and what should the discrepancy be? And with a totipotent progenitor, we're going to get high discrepancy usually, but 1 progenitor. And then we've got the one with discrepancy 0, but we might have many progenitors. And now we can sort of explore this trade-off and think about what makes sense in terms of the cell lineage trees we have. And that's what CARTA does. And I won't sort of tell you exactly how we do it, but it's, there's a fun combinatorial optimization algorithm that underlies it that Palash and Richard sorted out. So I'll just show you one quick example, and then we'll wrap up. So this is differentiation in mouse hematopoiesis. So this sort of older lineage tracing technology. They had sort of barcoded cells at day 0, just a single barcode insertion, and then they measure them at days 2, 4, and 6. So actually they get these weird lineage trees where they're all stars, but you have thousands of them. And then here's over these 130,000 cells. This is a 2D visualization of the gene expression. So these are the differentiated cell types down here that they have. And then they had a bunch of cells in here that were what they just called undifferentiated cells. Okay, these are like the progenitors, or who knows what they are, right? There's this big mass in the middle. Okay, so what we can do is run CARTA on this data. We can sort of explore this trade-off, and we find this sort of nice point right here where we get 7 progenitors with some sort of small discrepancy. And we ran a bunch of other methods that claim to sort of find these differentiation maps in different ways. This is a hematopoiesis, blood development. This is sort of the somewhat canonical differentiation map, and CARTA matches this the best. Okay? Some of these recent methods is called PhyloVelo. This is like doing some sort of phylogenetic velocity from RNA sequencing data in some phylogenetic form way. It gets this really strange thing that doesn't kind of make much sense. This is one of these distance-based methods that gets a binary tree. So here we're sort of matching this map the best. We can also, though, actually predict the fate of these undifferentiated cells by having predicted their progenitors. So we predicted which progenitor they are, and then that tells us where they should have ended up. And then we can see how well do we do by trying to just match in gene expression space alone, and we sort of get something that kind of makes sense. So that's CARTA. So derive this cell differentiation map from lineage tracing data, and this trade-off that's there between the two. And we get these interpretable cell differentiation maps. I showed you mouse. I didn't show you this other mouse embryogenesis data set that we're doing in collaboration with Michelle Chen, whose name was on the slide but I didn't explicitly mention. And then you could ask, what's next? Right? So I showed you actually two different technologies, spatial sequencing, lineage tracing. So why not do them both together? And this is the new thing that's happening, and this is spatial lineage tracing. There's only a couple of these data sets out there, but Ewan Ma, who's here, and Gary Hu, who's weirdly I couldn't find a picture of, he's not here because he's at ISMB, and he just today presented this paper. So if you had chosen to go to ISMB instead of this meeting, well, you'd be eating poutine tonight instead of pizza on the beach for one. So Montreal is where you'd be, so you probably chose wisely. But you would have heard this talk, and instead of hearing that talk, you heard my talk. So you probably lost on that deal. But this one is actually trying to look at spatial lineage tracing data. It actually becomes what's called phylogeography. So phylogenetics about sequence, phylogeography about sequence and space together. And we sort of pulled out this kind of really interesting thing in the data, which is to do this right, you had to think about the fact that when a cell divides, cell division is spatially symmetric. So we actually had this sort of spatially symmetric phylogeographic model, and you can read about that in the other paper. So that's it. I actually, by my clock, stayed under time. So thanks for listening, and I'm happy to take questions either here or on the beach. And I'll acknowledge the folks that did the work, and happy to take any questions. Thank you.